Let me start by adding my very warm welcome to Tim's. It's great to have so many people here and so many people interested in the area. It's a very exciting time in our field. It's a very exciting time in genetics, genomics, and its impact on healthcare. I hope you think that already, and if you don't think it already, I hope you think that in a couple of hours' time, having listened to the various presentations this afternoon. With my college hat on, as Tim mentioned, I'm lucky enough to be a fellow of St Anne's. It's great to be part of a college that has the vision, and critically the visionary leadership, to want to engage in partnerships with the university around some of the exciting developments and allow us to take on the broadest perspective to understand what their consequences are and to reach out to a much larger community in giving a sense of the developments and the excitement and the possibilities and also the challenges in the field. With my day job at the Wellcome Trust Centre for Human Genetics, it's great that from the Centre's point of view that we've been able to partner with St Anne's in developing the Centre for Personalised Medicine. We live genetics and genomics. We like to think that it will impact both scientifically and in the lives of individuals through healthcare and the way healthcare is developed. And the Centre gives us a great opportunity to collaborate with others in Oxford in the same area and to really reach out to a number of different communities to tell them about and give them a sense about and involve them in the developments in the field. So my job today is to set the scene. What I want to do is to do that aiming a talk explicitly at the non-specialists amongst you. Many of you, I think, will know much of the science that I'm talking about. I hope even you will find one or two bits of interest and novelty. But unashamedly, I want to try and set the scene and give a sense of the excitement and the developments in genomics over the last 10 or 20 years, and in particular the last few years, aimed at people who don't come from a genetics and genomics background. So in terms of genetics and human diseases, there's a spectrum, and at one end of the spectrum are diseases which are typically individually quite rare, often serious, typically serious, in which genetics is all of the story. So you'll know of conditions like Huntington's disease or cystic fibrosis, where the genetic story is that if you inherit in some cases one and in some cases two mutated copies of a key gene, then you will definitely get sick with the condition. Thankfully, they're individually rare. Collectively, though, these sorts of inherited genetic conditions affect about 1% of children born. I guess our progress in what we might think of as the modern era of genetics and genomics started 25 or so years ago with the identification for the first time of some of the key genes involved. So in 1989, the gene that was involved in cystic fibrosis was discovered. It's interesting and salutary and I think cautionary to ask, well, what have we done in the 24 years since then with cystic fibrosis? Until about a year ago, that genetic knowledge hadn't really been used to develop treatments. There's now a drug available that treats a small proportion of people with cystic fibrosis that makes explicit use of our genetic understanding, and that drug is extremely effective. But for most cystic fibrosis sufferers, the genetics hasn't yet led to a cure or a treatment. What it has done, and it's typical of many of the rare diseases where genetics is important, is that it's allowed identification of people who are ill at a much earlier stage. Genetics helps with the diagnosis. And for people born with cystic fibrosis in the 1960s and 1970s, the median life expectancy was about 10 years, and now it's around 40. So although genetics hasn't yet solved the problem for us, there are a number of interesting leads, it's helped with the treatment and in particular the diagnosis of individuals. And there are now thousands of different conditions, so-called Mendelian conditions, because the inheritance follows Mendel's rules, genetics is all of the story. There are now thousands of those conditions where the genes involved have been identified. I guess the next really big discoveries in genetics involve the genetics of breast cancer, where two genes called BRCA1 and BRCA2, you'll see the names of a number of genes in my talk, there won't be a quiz afterwards, they're usually pretty unmemorable. So BRCA1 and BRCA2 are breast cancer 1 and 2. Two genes were discovered in the mid-1990s where mutations in those genes, so particular sorts of changes in the genetic code in those genes, were responsible for very substantially increasing the risk of women carrying them to breast cancer. So, for example, individuals with a mutation, particular kinds of mutations in BRCA1, have got a lifetime risk of breast cancer of about 80%. For a typical woman or a woman without BRCA mutations, that lifetime risk is more like 10 or 12%. And for ovarian cancer of about 40%. So the discovery of those genes has been useful in situations where breast cancer runs in families, and where women are in such families, they can, in consultation with their doctors, decide, first of all, whether they want to get tested or not. The genetics of this particular inheritance for BRCA1 and BRCA2 is such that even if you're a woman in a family where individuals carry the mutation, you've got a 50-50 chance of carrying it or not. So half the people tested will get a negative result. They'll know from that that their risk isn't the elevated risk that other family members may have. They're back to the non-trivial risk, but rather lower risk that a typical woman would have. And where women choose to get tested and test positive, they can then think about various possible strategies for reducing their risk. And there's celebrated publicity around Angelina Jolie recently, who had that information and chose to have mastectomy as a consequence. So it's a case where genetics has helped with diagnosis, and you'll actually hear, I think, in Ingrid's presentation a little bit later, where genetics can help with treatment as well. So the next big advance in our field involved the Human Genome Project. If you're not familiar with it, what we mean by a genome is the entirety of the DNA, the instruction manual carried by an organism. So in the case of the human genome, we carry our genetic material in 23 pairs of chromosomes, and in our case, our genetic code is written in about 3 billion letters. Chemicals which make up the DNA. We can think of them, if we use the analogy of our DNA as being an instruction manual, they're the letters which give the cells in our body the instructions to do the appropriate things at different times. So it's tempting in our world to think of us as being rather more sophisticated than many of the other creatures on the planet, and we might think, and indeed we did think for many years, that as a consequence we need rather more DNA than they would. That's not strictly true. So salamanders, little lizards, have about 10 times as much DNA as humans do. There's broadly a relationship between the complexity of an organism and the amount of DNA it has, but it's not as straightforward as one might think, as is the case with many things in genetics. The achievement of the Human Genome Project, and it was a project that took 10 or 15 years and thousands of scientists working in many different countries, a total cost estimated at somewhere between 2 and 3 billion US dollars, the achievement of the Human Genome Project was to read one copy of a genome. We get one copy from our mother and one from our father, and we pass on one copy to each of our children. That happened with the first draft in 2001, the final version of the project in 2003. For those of you outside the field, there was, of course, a lot of fanfare at the time, which you might have been aware of. How do we think about it? I think the easiest way to think of the success of the Human Genome Project is it gave geneticists a map. You can think of our genome as a vast, unexplored territory. There are lots of bits of it that we don't understand, we'd like to learn more about. And before we had the human genome, it was like exploring Africa, for example, in the days before there were decent maps. It was quite hard to know where you were and relate findings that you might have to other findings that could be just down the road in ways that you'd be unaware of. So the Human Genome Project has facilitated a lot of research. It hasn't, I think, directly had the impact on health and healthcare that it might have, but many of the results and discoveries that have followed since have done, I would argue. So if you think of the Human Genome Project as focusing on what we have in common as humans, our DNA, there are also differences. So if you compare one of your chromosomes to a chromosome in the person sitting next to you, or indeed if you compare one of your chromosomes with your other chromosome, compare the one you inherited from your mother with the one you inherited from your father, then they'll agree at most positions. They'll differ on average at about 1 position in 1,000. And those differences are responsible for the genetic differences, the differences we display that are due to genetics. Things like how we look, how tall we are, aspects of our personality have a genetic component, and critically, the fact that some people suffer from some diseases and some from others also has a genetic component. Again, I told you the sobering thought about the salamander. Another sobering thought here when I said that if you compared your DNA to the DNA of the person sitting next to you, they'd agree at 999 places out of 1,000. So if, heaven forbid, but if the person sitting next to you were a chimpanzee, rather than the attractive, warm and interesting human being that they really are, if they were a chimpanzee, your DNA and their DNA would agree at 99 places out of 100. So we've actually got a lot of similarity genetically with our nearest relatives. So I said at one end of a spectrum in terms of genetics are the rare diseases, where genetics is all of the story. At the other end of the spectrum are kind of all diseases, and in particular the common diseases, where genetics is part of the story, We've known that for many years. If you take any of the common diseases, we've known for many years that they run in families more often than they should by chance. So that points to genetics being an important part of the story. But we also know that genetics isn't all the story. Genetics doesn't determine whether people get sick with these sorts of conditions. It influences it. And so it's nature, the genetics part, and nurture. But until recently, in spite of very substantial research efforts, we had made little headway in understanding the genetic basis of common diseases. Which bits of our DNA meant that some people were more likely to get heart disease, others were more likely to get diabetes, and yet others perhaps more likely to get arthritis or schizophrenia. That's changed over the last few years, and I want to give you a sense of that. So I'll show you various pictures like this one to orient you. Each one of these kind of stripy bars is a cartoon of one of the human chromosomes. They're ordered and, in fact, numbered by size, human chromosome 1, 2, 3, 4, and so on. And what I want to do in this next series of slides is to give you a sense of the pace of development since 2005. Now, had I been giving this talk in 2005, I wouldn't have been because it wasn't such an exciting field then, but had I been giving the talk, I would have been able to say that there were maybe a handful of examples where we knew about common genetic variants reliably associated with, so involved in susceptibility to common human diseases. That position changed in what we think of as a kind of new era, and I'll explain why the change happened. So this little lollipop here at a particular place on chromosome 1 signifies a research finding that showed that there was a change in the DNA letter at that point with the property that if your chromosomes had one letter, say an A at that position, compared to a T, you are more likely to develop a particular disease. The disease in question for this finding, and it's got a colour on its lollipop, is a disease called age-related macular degeneration, an eye condition that people suffer from as they get older. So I want you to think of a picture where before this slide we had a handful of examples where we knew about genetic variants associated with disease. There was a new discovery in 2005. There are a few more in 2006, another one for macular degeneration, and a number for Crohn's disease, a form of inflammatory bowel disorder. And then the position started to change, so that's what it looked like at the end of 2007. Quite a lot more discoveries, 70 or 80 altogether, across a range of different diseases. That's the position 2 years later in 2009, 2010, and 2011. From having a handful of examples in 2005, we now know hundreds of examples of diseases for which we know thousands of positions in our DNA where changes in the letter in our DNA code affect your chances of developing that disease. There are many, many different diseases involved. Here's a list. You can't even read it. It won't be on the quiz, so you don't have to worry too much about that. But there are examples like breast cancer, heart disease, ordinary human conditions like height and weight. Figure here, we understand the genetic basis of those better than we should have, diabetes, schizophrenia, and so on. I showed you the picture of 2011. That's a slightly later position where the genome is now covered with instances where we know particular letters that affect people's susceptibility to diseases. The picture started to change in 2007, and we were very lucky in Oxford to be involved in some of the major studies that led to those developments. So each year, Science magazine is one of the two main general science magazines, the other one's Nature. And each year, in a rather pompous way, science kind of stands back from its whole sphere and picks the area of science which had the most exciting development during the year. And in 2007, it was this rapid explosion. Well, it seemed like an explosion then. We look back on it now as the kind of tip of an iceberg. But it was this rapid explosion in our understanding of the genetics of common human diseases that science picked as its breakthrough of the year. I want to give you a little sense about how we do these studies. It's not rocket science. Well, it's not rocket science in two senses. It's not rocket science because it's transparently biology rather than astrophysics. It's also not rocket science in the colloquial sense that the basic idea behind the studies isn't very hard. So I want to share that with you. What we do typically is to take a large number of individuals, think thousands, and these days actually for some diseases, tens of thousands of individuals who have a disease of interest, say heart disease, and a similarly large number of people who don't have the disease. We call them controls. They typically be samples from, individual sample from the population. And what we do is to measure their genomes at many places, 500,000 or 1 million places, which we know in advance vary between different chromosomes. And then the signal we're looking for are places in the genome where there's a particular letter, maybe the two variants of that position, there'd be an A on some chromosomes and a T on others. We look for places in the genome where the A is more common in the sick people and the T more common in the healthy people. Well, why is that? Because if you think about it the other way around, suppose it were the case that if you had the A, you're more likely to get the disease, then A would be more common among sick people, because at least some of them will have developed the disease because of that change. So it's the ability to do these large studies on thousands of individuals, and these days tens of thousands of individuals, relatively cheaply, which has allowed the explosion I just described in our knowledge of places in the genome associated with susceptibility to disease. Why do we do these sorts of studies? I think most of us would say for two different sorts of reasons. And for most of us, the first reason is more important than the second one. And the first one's the following. For many human diseases, in spite of huge amounts of research, often over more than 100 years, we know depressingly little about what's causing the disease. We know depressingly little about the biological changes inside a person which trigger heart disease or schizophrenia or diabetes or arthritis. Genetics gives us a whole new foothold into that. Suppose we do find that if you have an A in this position rather than a T, you're more likely to develop heart disease. What we can then do is look biologically and find out what's going on biologically in people who have the A compared to those with the T. And when we find the difference and reliably establish that difference, that gives us a pointer. That's a biological change where having, for example, more of this gene working leads to heart disease. And a striking feature of the thousands of discoveries that I showed you on those cartoons is that in the vast majority of cases, although in very few cases do we know exactly what the biological mechanism is, but in the vast majority of cases they seem to be pointing to entirely new bits of biology that experts in the particular disease would not have guessed was important for the disease. So for many of us, the main reason for studying genetics is that it gives us a set of tools to understand the diseases better. And if we understand the diseases better, we have the possibility of developing new treatments or new drugs and so on. So that's, as I said, I think that's the main reason, and you'll get more of a sense of that as the afternoon develops and its consequences for healthcare. It's also the case, so if you know some variants make you more likely to get heart disease than others, you can measure those variants in people. And you could then say, well, they've got an A here rather than a T, that'll increase their risk by this much, and they've got a G there rather than a C, that'll increase their risk. You could use this to get better predictions of the risk for an individual developing the disease. Now, I think most of us in the field would say that's currently less valuable. And the reason is that although we've discovered lots and lots of changes in DNA that affect your risk of disease, individually, they change your risk only slightly. So they're real, and they can point to important biology, but maybe if you have an A rather than a T, it increases your chance of getting heart disease by 5% or 10%. So for most people with these common variants and a typical disease, some of them you'll be better off and some of them you'll be worse off, so they won't have a big effect on your risk. As the genetics goes further, and the example of breast cancer that I talked about earlier is one where we're already this far, we expect to discover risk variants or collections of risk variants which are better at predicting and identifying people at increased risk. But by and large, although there'll be some discussion of this later this afternoon, we're not there yet. So I told you how we do the studies. We're very lucky, as I said, in Oxford in contributing to the largest of the initial studies, and indeed many of the studies which have followed. And in our scientific world, we get rather excited when we think we've done something worthwhile and one of the top journals likes to publish it, which they did. It's much scarier for a scientist when the press get involved. They did here, which was great. So The Independent, a newspaper I now think rather fondly of, devoted its front page and actually all of its second and third pages to these discoveries. Although in a kind of tantalizing way, they thought our advances in genetics should share their front page with Tracy Emin, whom many of you will know as one of Britain's more avant-garde modern artists. The Daily Telegraph, also very exciting. So Tim didn't mention, you may be able to tell from my accent that I'm Australian rather than British, although I've lived in this country for a long time, but still share my Australian sporting loyalties. So here we were on the front page of the Daily Telegraph, shared with an exciting picture of English footballers. So what was going on in June 2007 that warranted the front page of the Daily Telegraph for football? So England had just reached the dizzy heights of beating Estonia in a European qualifying competition under Steve McLaren, which they then went on not to reach in any event. So I want to show you one or two examples of the data from these studies. And remember, I've said what we do is we take sick people, healthy people, we measure them at lots of places in their genome, and we look for differences. So here's probably the most interesting finding from our initial study, and a number of others reported at the same time. So each one of these black dots is one of these positions, an A or a T, that we measure. And as we move across the x-axis in the picture in this direction, we're looking at different places on one of the human chromosomes, in this case human chromosome 9, and from here to here, there are about 2 million positions, letters along chromosome 9. And what you see, so the distance off the axis, so the distance up here, is a measure of how strong the statistical evidence is for there being a difference in frequency between the sick people and the healthy people. And what you see here and here is kind of background noise, nothing very exciting happening. And then here in the middle, there's a huge jump in the evidence, as there are letters that we're measuring which are much more common, relatively more common in the sick people with heart disease than the healthy people. In fact, having said that most of these variants have a relatively small effect on risk, each extra copy of whatever letter it is that we're measuring here, each extra copy of that increases your risk by maybe 30 or 40%. So if you have 2 copies of that variant, the effect on your risk for heart disease is comparable to the effects we worry about with cholesterol. What would we do when we have this kind of discovery? We'd look in that part of the genome and say what genes are there. So genes are the bits of the genome which make proteins. There are 2 genes in this region called deeply unmemorable CDKN2A and 2B. They were not on anyone's radar in terms of heart disease. And actually, although this discovery is now 6 years old, we still haven't got to the bottom of the biological mechanism. So these genes weren't on the radar of anyone involved in heart disease, but they were very well known to cancer biologists because they play important roles in the cell cycle. We don't yet know how that fits in with heart disease, but it's an important clue to understanding the disease better. So that was perhaps our most interesting single finding. Here was the one which attracted the most attention. So this is a result we found in our study of type 2 diabetes, the type of diabetes that affects people in late middle age, typically. We were working with a number of other groups who were also studying type 2 diabetes who didn't find this. So that was a bit disconcerting for a while. And then when we looked more closely, it turned out, so weight or obesity or body mass index is a risk factor for diabetes. People who are overweight are more likely to develop diabetes. So you might think if you're studying the genetics of diabetes, you should worry about that. The other 2 groups had done that. They'd done clever things. One of them had only studied diabetics who were thin, and the other group had carefully matched their diabetics with their controls to take weight out of the equation. Our researchers in diabetics hadn't been clever enough to do either of those things, but it paid off. We found this result, and in fact, on closer examination, it wasn't a result, it wasn't a genetic variant that was directly driving susceptibility to diabetes, it was driving weight or body mass index. So people who have 2 copies of whatever this letter here is are on average about half a stone heavier than people who don't. And it was the first finding, and with a fairly substantial effect for weight. As you can imagine, the press got very excited. Oops. The press got very excited with that, and the headline writers had something of a field day. Even the Financial Times, that doyen of serious British reporting, got interested, both actually with their front page and encouragingly for us with their editorial. They were very positive, as we are, about the fact that we were just announcing these results. We weren't trying to protect any intellectual property in them, but this is by far my favorite of the headlines. So why has all this change come about? What's brought us here and you here? Well, it's a number of different things. There have been dramatic changes in our technological ability, our ability to measure DNA, and I'll come back to those. Advances in our ability to analyze these large amounts of data. My own background is in statistics, so the skill set I bring to these studies is in part, or myself and my research group, is in part the ability to extract information from huge amounts of genetic information and healthcare information, and also in terms of basic biology. So here's a graphic which gives you a sense of the cost of measuring DNA. Remember I said that in the first version of the Human Genome Project, it cost about $2 or $3 billion to measure one human genome. So these are the costs of measuring a megabase, so 1 million letters in the DNA, and you'll have seen that they've gone down by a factor of about 100,000 over 10 or so years, and critically over the last 3 or 4 years, or more accurately, from the 4 years from the end of 2007, the costs went down by a factor of about 10,000. They've kind of leveled off a bit, disappointingly for us in the field, but there's been a dramatic change. So it's just much, much cheaper now to get the genetic information we need to do the kinds of studies we're describing. There's the biblical adage that we should all know ourselves, or that you should know thyself. So I don't think the Bible quite intended it this way, but if one were to think in terms of our DNA, we can know ourselves rather more cheaply now than used to be the case. So, as I said, it took about 10 years, thousands of scientists, and $2 or $3 billion to read one copy of the human genome in the Human Genome Project. So now one of these DNA sequencing machines, and we have 6 of them in the Wellcome Trust Centre up the road, they can read an entire human genome in about 24 hours. Current costs are about £2,500 or £3,000, and it's anticipated that within a few years it'll be about £1,000. So critically, the cost of reading an entire genome, someone's entire DNA information, is getting down to the sorts of cost levels of many other medical tests. And actually, so there are bits of genetic information which are measured routinely now for people, for example, with family history of breast cancer. Reading the DNA in a single gene in the NHS currently typically costs £300 or £400 or £500. So we're not far away from being in a situation where reading a whole genome isn't much more expensive than a number of current genetic tests. So there's a huge challenge, as I said, on the informatics side. So this is a series of, actually made up letters, but a series of letters that represent 1/1,000,000th of one genome. And if you think about measuring genomes in thousands of people with a disease and thousands of people without a disease, so you've got 1,000,000 times more information than that in each individual and thousands of individuals, then you'll get some sense of the challenge of looking for the patterns and the similarities in the sick people compared to the healthy people. And as we talk about trying to translate that information into clinical healthcare, there are a number of issues or challenges, illustrated by the cartoons, and it's the kind of thing we'll explore as we go on, of making that information accessible to the clinicians who have to use it to make decisions about treating patients. So how does this lead to personalized medicine? I want to give you a number of kind of little cameo examples of settings where it can and will make a difference for healthcare in the short term, and then they'll be explored rather more after the coffee break by Ingrid and Gabe and Chris. So the first example is an area called pharmacogenomics. So many aspects of our physiology are affected by our genetics. One particular aspect which is affected by genetics is the way we react and metabolize drugs. So how quickly the drug goes into our bloodstream varies between people in ways that have, at least in part, a genetic basis. And whether or not people react to a particular drug adversely differs between people, and in some instances, some or all of those differences are due to genetics. So as we understand those aspects better, we could try and tailor the way we give drugs to people. If we knew that someone metabolizes a drug more quickly, so it goes out of their system more quickly, we could adjust the dose accordingly. So genetics could be used to tailor choices of drugs or dosages. And also there are instances, and I'll give you one example, where we know about genetic factors which affect side effects. So there's a drug called abacavir, which had been quite successful as a so-called antiretroviral drug to treat HIV or AIDS. It's a drug which was developed, and in the late stage of trials, it turned out that about 5% of people had a very serious adverse effect, something called hypersensitivity, which was at least very serious and then sometimes could be fatal. So it takes, as you'll know, billions of dollars to get a drug to that stage, and then you find out that there are a small proportion of people who are adversely affected. The only thing you can do is not use the drug. So all of that research and a drug which works really well for 95% of people is then useless, unless, as was the case here, it's possible to tie the adverse reaction to genetics. So it turns out here that there's a particular genetic variant, something called HLA-B5701, which occurs in about 5 to 7% of Caucasians, and it was individuals carrying that variant who suffered from the adverse side effect. So armed with that information, you can now do a genetic test, first of all, check to see whether your patient has 5701, and if they don't, prescribe the drug, and if they do, use something else. So a drug which worked well for most people but could not be used on any because of the adverse side effect gets rescued, and one's able to use it fairly broadly because of the genetic knowledge which allows us to identify in advance individuals who will suffer the side effect. That's one example. So I want to talk briefly about cancer. So however you currently think of cancer, I want you to adopt a slightly different perspective. Cancer is fundamentally a disease of the genome. So, roughly speaking, all the cells in our body have the same copy of our DNA, the DNA we inherited from our parents. The exception to that, and it's critical, are cells that are cancerous, that are in a tumor. You can think of those cells as cells with screwed up DNA. Stuff has gone wrong with their genome. So if you think of our genome as our instruction manual, in cancer cells, things have gone wrong with it, typically, which either causes the cells to proliferate more than they should, or causes them not to die when they should. But the end result is that they grow in mass and create tumors. So what goes on in cancer is it's a set of cells that have got messed up genomes. So now, for the first time, it's possible to measure the genomes in the cancer cells and work out exactly what's gone wrong in those cells. So that can be extremely helpful from a research point of view in understanding cancer better. It can also be really helpful in targeting treatment to the particular changes in the cells in an individual tumor. So I want to try and give you some sense of the changes in a cancer tumor. So what would I like to do? I'd like to pick an individual who has cancer, measure their ordinary genome, the genome that's in most of their cells, so that would be 3 billion letters, and then take some cells in their tumor and measure their genomes and show you how they're different. So it'll take us a while to go through the 3 billion letters in the normal cells and the cancer cells. So here's a way of displaying that. So how do you think about pictures like this? So the inner circle tells the story of large bits of chromosomes that are not where they should be in the tumor cells. So there's a large chunk of chromosome 3, which should normally be on chromosome 3, that's kind of unattached itself from there and is stuck on chromosome 18. Or some large chunk of chromosome 3 is in a different place from where it should be in chromosome 3. So in this particular cell or set of cells from so melanoma is a skin cancer, from a skin cancer tumor, there were about 37 examples of these gross changes in the genome. There were another 66 examples illustrated around here of quite large bits of DNA that were that should be there and were missing, or that should be there once, but were there more than once, 2 or 3 times. And on top of that, there were about 33,000 examples of individual letters in the DNA which were changed between the cancer genome and the ordinary genome in that individual. So I want you to get a sense, when I talked about cancer cells having very messed up genomes, they're very different typically from the ordinary genomes in the individual. They're also quite different across individuals with the same kind of cancer. So I'm going to show you 6 pictures now of the same kind as the ones I've just shown you for the genomes of the cells in breast cancer tumors from 6 different women. And all I want you to notice is how different they look. So whereas up until now, typically in cancer, we've classified cancers according to which part of the body they fall in, skin cancer, lung cancer, colon cancer, breast cancer, and so on. Actually, we can now read the genetic information and start to think about classifying cancers in terms of the genetic changes which are driving the cancer. In breast cancer, that's been the case for a while, not through the ability to read whole genomes, but to read aspects of the genetics of breast cancer. So if you've heard about discussions of breast cancer which talk about HER-positive tumors and so on, that's because of particular genetic changes which we've been able to measure with existing technologies. We'll now move into an era where we can measure the whole genome. That could make a difference because, in principle, we could then target drugs not generally, but specifically at the changes in an individual tumor. And here's a really encouraging example of that general principle. So to go back to melanoma, as it turns out, many, but not all, about 2/3 of melanomas have a particular change. So BRAF is another gene, it's an important gene, and this is kind of genetic shorthand that says at a particular position in the protein, the 600th as it happened, one amino acid replaces another one. So informally, you can think of that as a change in the cancer cells which leaves this gene BRAF turned on much more than it should do. So it's producing stuff that it shouldn't normally produce, and that's what's driving the proliferation of the tumor. So that was discovered about 10 years ago, and of course people had the idea, why don't we try and develop a drug that deals with that? So again, saying at a very informal level, you're producing too much of this BRAF stuff, so you'd like to produce a drug which kind of mops it up and stops it from doing what it would otherwise do, which is to encourage proliferation. So that's called an inhibitor. And I'm going to show you pictures from an early trial now a year or so ago of individuals who had very advanced metastatic melanoma. So this is a scan of an individual, and I don't know too much about these things, and you don't need to know too much either, but with the exception of the brain and the bladder, all this coloured stuff is really bad news. That's all tumour that spread from the initial skin cancer tumour throughout the body. So this is one individual in the trial that had this kind of change, and they were given this BRAF inhibitor. What happened? That's the scan 2 weeks later. There was an extraordinary change in the ability of this drug targeted at the particular changes in that tumour to basically remove almost all of the metastatic tumours. Now, this is very encouraging. There's effectively no current treatment for melanoma, but cancer is quite clever. If I showed you the same picture 6 or 8 months later, then the cancer would have found a way of getting around the drug. But I think there's optimism, and there are others here who know much more about it than I do, there's optimism that these kinds of ideas will lead to important developments in the treatment of melanoma. But it's one example of being able to tailor a drug at the specific changes in the tumour by measuring what's going on in the tumour. My final set of examples have to do with reading entire genomes. So we've been involved at the Wellcome Trust Centre in a collaboration with Illumina, the company that make the best current generation sequencing machines. And what we in Illumina were trying to do was to work out which bits of clinical medicine had the property that DNA sequencing of entire genomes could make a difference in the short term. And our idea in doing this experiment was actually to try lots of different things, lots of different clinical conditions and types of conditions, so that we could stand back afterwards and kind of evaluate the impact of clinical sequencing. I'll give you just one example. Early on in our study of a child who had major issues with the shape of her skull and also her brain development, something called craniosynostosis. She was very affected, and we had hypothesized that the most likely explanation here was that she had inherited a single change in an important letter in her DNA by chance from either her mum or her dad that was causing these differences. So here's a picture of the girl, and you can see her head is not the shape that you would normally expect, and that's a scan, and most of our skulls are more solid than that. So these are serious issues. So we did the sequencing, and we were able to identify a particular position in an important gene which we think is responsible for the condition. That attracted a lot of attention. It was one of the first examples of sequencing entire genomes, which had an impact on clinical medicine. It wasn't in this case, and a number of other examples like this, it wasn't in this case that the doctors could then say, I know how to fix that. What it did do was to identify what the likely cause was. It was extremely helpful for the doctors in giving advice to the parents about the risks for having subsequent children. So there's some genetic conditions, depending on the mode of inheritance, where if one child has it, there's a 1 in 4 chance that subsequent children will be affected, whereas we're able to say here that because it was a freakish kind of one-off change, it was extremely unlikely that subsequent children would be affected. It actually had a really important pragmatic effect. The girl was a little bit older by the time the study was done, and the local education authority needed some convincing that there was she somehow or other needed special treatment and special needs for education, and being able to say, we've seen the change in the genome that causes, or is likely to cause the problem, made the difference there. So that's one example. As I'll go on to say, our study of 500 individuals has kind of morphed into a much larger study within the UK. But before doing that, one final point. So I've talked up until now about the genetics of us as humans. All creatures have genomes, and in particular, the organisms, the bacteria and the viruses that make us sick, they've got genomes too. And those genomes are typically much smaller than ours, so they're much easier and quicker to read. And so if we think about the way clinical medicine deals with bacterial infection, for example, it's now possible, and there's a leading program in this in Oxford, it's now possible to take the bacteria that's infecting someone and just read its DNA sequence. What does that allow the doctor to do? It instantly confirms exactly what it is that's causing the infection, or if there is more than one thing, identify that. Just by looking at the genome, it'll be possible, as we move forward, it'll be possible to work out which antibiotics will work and which ones won't for that particular infection in that patient, rather than trying one and seeing whether it works and then trying another. And for infectious agents where the spread of the infection is important, so for example, hospital outbreaks of C. difficile or MRSA, you can use the genetics as a fingerprint to trace the spread of infection. You can work out from reading their genome that the woman in this bed in this ward has exactly the same bug, and so was probably infected either from or by a common source from the man in the bed in the ward next door or whatever. So in real time, hospitals will get much better information on likely routes of transmission in hospital outbreaks of C. difficile or MRSA and so on. So genomics, I just want to make the general point that genomics will have a big impact in terms of bacteria, over and above our ability to read human genomes. And I'll just finish by telling you about, or reminding you about if you're already familiar with it. So I talked about our project in Oxford to sequence 500 genomes of individuals to see what its impact on healthcare was. I think in part because of the success of that project, but for a number of other, all good reasons, I think. Just under a year ago, the Prime Minister announced a huge study in the UK to sequence the genomes of 100,000 people within the NHS, to start using genetics, to get the NHS ready to use genetics, and to start using that to affect clinical care. So, as with our study, that'll initially focus on a couple of categories of diseases. Cancer is one example. Patients with rare diseases, like the young girl whose picture I showed you, is another, and on the genomics of bacteria. So I'll finish with that. What I've hoped, or tried to do, and hope I've had at least some success, is to give those of you who aren't specialists in the field a sense of the extraordinary pace of change in our field and the possibilities that have been opened up to use genetics and genomics to affect, and in particular to personalize healthcare. Thanks very much.